Awesome. So I'm hoping that you guys survived the background section pretty decently. Essentially, what I want you to really think about and take away from background or the background lecture is what are enzymes? Why do we care about enzymes? Why are NAD and FAD important, right? And that's largely going to be because they're shuttling hydrogen, they're shuttling electrons. Super important, and you'll see why in this lecture specifically, right? NAD and FAD shuttle electrons and they move around hydrogens. And then you also need to understand really kind of the concept of a cell membrane and how cell membranes kind of keep things separated and how they kind of function literally like oil and water, fat and water, right? Like they separate out different areas from each other, and some in order to transport across one side to the other side, it needs some sort of either gradient, which we'll talk about today, or it needs some sort of component of active transport, which we'll talk about today. And really, the other thing that you need to understand from that lecture is what are endergonic and exergonic reactions and what are coupled reactions. So, endergonic means that they're energy consuming, they require a net input of energy to proceed. Exergonic reactions produce energy. So if we're trying to create and make energy available for our human body, we need to kind of have more exergonic reactions than endergonic reactions, or we need to balance out how many reactions are going to be consuming energy when we're making energy, right? Because you can't really technically make energy. You can kind of just shuffle around energy and pull energy from other areas. Energy, just like matter, can't be created or destroyed. So really today we're going to be talking about bioenergetics, or how we're getting said energy, how we're getting said ATP. Here's kind of the primary goals or the primary topics that we're going to be discussing today around this lecture. I mean, I guess you could technically be listening to me moan on and on and on, or I guess groan on and on and on, which would be probably punishment for you. We're going to talk about ATP, what it is, how we store it, how we use it. We're going to talk about how we kind of generate ATP via phosphocreatine, the glycolytic system, and the oxidative system. And then something that I want you to kind of be thinking about as you're going through this is that while we're going to talk about each one of these in isolation, they're not running in isolation. They have different kind of contributions and percentage efforts as they're moving in our body, but they're not running in isolation. Each one of these is always going to be working to produce energy or to kind of generate energy. I'm going to use produce a bunch, but to kind of produce the ATP, if you will. So what is the phosphagen system? So the phosphagen system is going to be the simplest and most rapid method of producing ATP, or the simplest and most rapid method of producing a high energy molecule. How that's going to work is you're going to have so hard pause. Let's talk about this really fast. Sorry. Arrows denote which way the reaction is going. So if you have arrows going one way, that means the reaction is going from, like in this case, if you're reading it this way, it's going from right to left. If you're reading it this way with this arrow, it's going from left to right. So it's being read reactants, products, or reactants and products. Complicated, but what this means is that this reaction is interchangeable. It can go either way depending on what's present, and that's ultra important. So over here, you've got phosphocreatine. So you have a phosphocreatine. You have a phosphate that is attached to a creatine. You have a PCR or CRP molecule. That phosphate, if you're going that way, can get donated from ADP, and it can shift from phosphocreatine to ADP to create ATP. ADP stands for adenosine diphosphate. So it means it has an adenosine molecule to it, and then it has two little phosphates as a tail. The goal is to create a third phosphate onto that tail, which makes it a much higher potential energy molecule. This reaction is going to be mediated by the enzyme creatine kinase. Remember, kinases means that it's having something to do with a phosphate group. First lecture, we talked about a couple of key enzymes. Kinases specifically deal with phosphate groups. And if our energy currency is really ATP, then those kinases are going to be super important because they're going to be shifting around that final high energy phosphate from other molecules and onto ATP, or pulling it off of ATP and releasing some of that energy. The phosphocreatine system, think of this as like your quick hitting, your really fast producing energy system. Think 40-yard dash or really that's like the high end, right? So it's the first really 5 to 6 seconds of just massive burst of energy. It's going to be the phosphagen system being the primary contributor. The other two, glycolytic and aerobic, are going to be kind of spinning up, but phosphagen is really the primary mover here. Since I said the word creatine, it kind of generally brings up the concept of creatine supplementation. You'll get into this quite a bit more in your nutrition class, but just as an aside, I want you to know that creatine supplementation does seem to work according to the science. Essentially what you're doing is you're super saturating your muscle with creatine. So if you go over to here, if you are increasing the amount of creatine that's inside of the muscle, you are essentially increasing the likelihood of it becoming phosphocreatine. So it's almost like a reserve tank, if you will. You're increasing the potential for a high potential energy molecule. You're increasing the likelihood of having a high potential energy molecule, if that makes sense. So it does seem to work. The research is kind of split. There's high responders and non-responders. For the most part, most people tend to respond fairly positively to creatine, and most importantly, it is quite safe, or at least it's been deemed to be safe by the majority of research. So glycolysis is a more robust pathway than phosphocreatine system, largely because it creates some ATP, but it creates NADH. And why is NADH so important? Well, it's giving us an avenue of adding hydrogens and electrons onto NAD to create NADH. So it's allowing us to offload hydrogens and offload electrons, creating this high potential energy molecule, NADH. Remember, super important. Keep that in the back of your mind that here we're creating NADH inside of glycolysis. It's important, I promise. We'll talk about it eventually in this lecture, I promise. Essentially, what glycolysis is doing is it's taking sugar, it's taking glucose molecules and breaking them down into kind of two molecules of pyruvate or two molecules of lactate. One is going to be fated towards kind of like the end of glycolysis and the end of energy production here. The other molecule is going to be aimed for continued energy production. So lactate is kind of the end, symbolizes the end of glycolysis, whereas pyruvate symbolizes really the start of the next energy system. Glycolysis occurs inside the cytoplasm of the cell, so it's not occurring inside the mitochondria, the powerhouse, remember, but it's actually occurring inside that fluid that's kind of sits inside of the cell membrane. Really, when you're going through these next couple slides as we're highlighting glycolysis, there's really two distinct phases. There's going to be an energy investment phase, and there's going to be an energy generation phase. So this kind of continues what we've been talking about in that if you're using an endergonic reaction to generate an exergonic reaction, you're essentially using coupled reactions, right? One reaction is driving the next, you can kind of think of it like stairs, if you will. Like as you get going, it gets easier and easier and easier. The net production of glycolysis at the end, the net production is 2 ATP and either 2 lactate or 2 pyruvate. Those 2 ATP, as you'll see, you actually produce 4 ATP, but because you're generating 4, you actually had to put in 2 ATP. And we'll talk about that, but again, it's coupled reactions, right? It's endergonic, energy consuming reactions generating energy producing reactions. This looks super complicated. In some ways, it really truly is overly complicated. But what I want you to start paying attention to on this slide and really on the next couple of slides is that here you have a glucose molecule. And so glucose is C6H12O6, right? So you can see essentially you have 6 carbons. Each one of these points is a carbon. So you have 1 carbon here denoted by 1 carbon here denoted by the number 2. This would be your 3rd carbon here, 4th carbon here, 5th carbon here, and then your 6th carbon is going to be here. The very first step of glycolysis is you take and you turn this glucose molecule, this C6H12O6, into something different. So you can see you put in ATP into this transformation reaction and you create glucose 6-phosphate. And since we go from ATP to ADP, we realize that we're actually losing a phosphate because we're going from adenosine triphosphate to adenosine diphosphate. So 3 to 2, triphosphate 3 to diphosphate 2. And the enzyme that is doing this is hexokinase. So hex means 6, kind of like hepta would mean 7. Hexa is the root for 6. And kinase, if you remember, means that you're dealing with a phosphate group. So the very first step of glycolysis is glucose to glucose 6-phosphate, or G6P for short, and we're catalyzing that reaction via the enzyme hexokinase. So hexokinase is making that reaction proceed easier or with less energy. You then have glucose 6-phosphate, or G6P, becoming fructose 6-phosphate. And as you can look, you're actually changing the structure of this glucose 6-phosphate into a 5 ring, or into a different shape ring, if you will, right? However, what you also need to realize is that you're not changing the number of carbons, you're just changing the shape. So you're not changing the net, what is there. You're not changing how many carbons are here. You're just changing where they are located. So you're rearranging them, or you're isomerizing them, or you're making a different isomer. So interestingly enough, glucose 6-phosphate and fructose 6-phosphate contain the same things, they're just rearranged differently. Kind of like you've probably heard of the concept glucose is the same thing as fructose or fructose. They're both simple sugars, and they have the same equation, they're just structured differently. And if you don't believe me, we can count. We've got 1 carbon here, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbons, right? So phosphoglucose isomerase is going to be the enzyme that's responsible for catalyzing this reaction, or making it easier to proceed. So that's glucose 6-phosphate to fructose 6-phosphate via phosphoglucose isomerase. Sorry, I keep hitting the button on my keyboard. From F6P, or fructose 6-phosphate, whichever one you want to call it, you're going to go to fructose 1,6-bisphosphate or diphosphate. You'll see them interchanged quite often. And again, you're catalyzing this reaction via the enzyme phosphofructokinase. So what we know is that kinase means we're dealing with another phosphate. And if we're going from fructose 6-phosphate right here to fructose 1,6-bisphosphate, what are we doing? We're adding in one more phosphate. And that's exactly what's occurring. So if you look here, PO3 is phosphate. You can see that we have this carbon ring that looks very similar to this carbon ring. The only difference is we have an additional phosphate group that's being tossed onto here, right? And that's happening because of phosphofructokinase. And if you look again, if we're adding a phosphate, what does that mean? We're having to get a phosphate from somewhere, and it's actually coming from ATP. So that means that the reaction that is going from fructose 6-phosphate F6P right here to fructose 1,6-bisphosphate is actually requiring an energy input. So it's endergonic. This ATP is donating its high energy phosphate group to make sure that this reaction occurs. Okay? From here, we're going to split, and we're going to really generate two different molecules. And that's really what you're going to see in these next two slides, if you will. I don't know which one is going to be easier for you guys to visualize. I put in several different versions of the same slide just so that you can see what they look like. So you're going to actually take this fructose 1,6-bisphosphate and you're going to cleave it essentially in half. Okay? And when you cut it in half with aldolase, you're going to create a 1-glyceraldehyde 3-phosphate, and you're going to create 1-dihydroxyacetone phosphate. Okay? Just trust me on this. So you're going to have this 6-carbon molecule that is fructose 1,6-bisphosphate that has a phosphate group here and a phosphate group here that gets divided in half, and it becomes glyceraldehyde 3-phosphate. If you look, 1 carbon, 2 carbon, 3 carbon, 1 phosphate. And then we're going to have dihydroxyacetone phosphate over here. And that is 1 carbon, 2 carbon, 3 carbon, and 1 phosphate. The only difference is look at the structure. The structure of these two is different, but they have the same constituents. They have the same components. So we have to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. The enzyme that's going to be responsible for changing the shape, not the molecules, but the shape, is going to be an isomerase, because we know that isomerases create isomers. They manipulate the structure, but they don't manipulate the things that are there. They don't manipulate the components, just the shape, right? And that's going to be triose phosphate isomerase. After dihydroxyacetone phosphate goes through a reaction with this triose phosphate isomerase, you end up with 2 glyceraldehyde 3-phosphates, right? And I think that this next slide makes it a little bit easier to visualize this. So if you look where everything's the same here, we're just kind of, rather than going across this way and looking at structures and stuff like that, I just have us going vertically and looking at names. If you want to look at structures, they're still there. It might help you, but for now, let's just stick with this. So off of 1 glucose molecule, we go glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate via phosphofructokinase. We then go from, I lied. So if you look here, you can see that we go from glucose all the way down. We split into two different things where we have actually one glucose has become two glyceraldehyde 3-phosphates. So you get 1 6-carbon sugar of glucose that gets changed into fructose, and then you add a couple of phosphate groups and you split them. And now you've got two different columns essentially proceeding. So I want you to imagine that this column right here is actually doubled. So there's this column going on, and there's another column going on that looks exactly the same. And that's supposed to be what this 2 right here is denoting, is that you actually have 2 glyceraldehyde 3-phosphates. And if you look here, you're kind of creating an NADH. And what do we know about NADH? We know that it's a high energy electron and hydrogen ion transporter. We know that we're essentially donating electrons and hydrogens here to create NADH. And that's kind of what's going on. And you can see that we actually are going from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. We're from a lot of people call this 1,3-diphosphoglycerate. You'll see it abbreviated 1,3-DPG. It's the same thing. We then have phosphoglycerate kinase. A kinase means we're dealing with a phosphate group again. And if we have 1,3-bisphosphoglycerate and phosphoglycerate kinase, the outcome, the product, is going to be 3-phosphoglycerate. So we're removing, I can't find my cursor, sorry. We're removing this 1. This 1,3 denotes that there's a phosphate on the 1 carbon and on the 3 carbon. And essentially here we're removing one of those phosphates, and we're just seeing a phosphate on the 3rd carbon, if that makes sense. So we're going from 2 ADP. We're dropping off 2 phosphates here. We're going from 1,3-bisphosphoglycerate to 3-phosphoglycerate. And those 2 phosphates are going to ATP, and they're going from ADP to ATP. So now we've created our first ATP. The first half we were energy consuming. We actually consumed 2 ATP. Now we just produced 2 ATP on this step, right? So now we're at net zero. 2 consumed, 2 produced. From here, we're going to move the carbon from we're going to move the phosphate from the 3rd carbon to the 2nd carbon. So we're going to go from 3-phosphoglycerate. We're going to use phosphoglycerate mutase. So we're actually going to shift it. So we are going to change the chemicals somewhat. And we're going to create 2-phosphoglycerate. And we're going to do that twice, because remember, we have 2 chains running during this. From here, enolase is going to serve to remove some water. And then from this reaction of phosphoglycerate with enolase, we're going to create phosphoenol pyruvate. So you can see that enol actually ends up coming into play here. So it's one way that you can remember what this step is. And then kind of the final reaction, if you will, is we're going from phosphoenol pyruvate, or PEP for short, PEP, to create pyruvate. And that's catalyzed via the enzyme pyruvate kinase. So we're actually, here we're pulling out those final 2 ATP, and we're taking the phosphate group off of this PEP, adding it to 2 ADPs to create 2 ATPs, and thus we're creating 2 pyruvates. It sounds overly confusing to go through it just like that. And I understand that. Unfortunately, it's just one of those things that you gotta go through 100 times to memorize, to learn, to understand. You need to use me. You need to set up meetings with me. This is, unfortunately, something that you have to know. So you will need to know for the tests, you will need to know what steps are energy consuming, what steps are energy producing. So where ATP is going in, where ATP is coming out on the second half, you need to know the molecules, and you need to know the enzymes. I don't care if you know the structures. I don't think you need to know the structures. I like when I was going through undergrad, we had to draw all this out. I don't think it's necessary. What you need to know is you need to know enzymes, you need to know molecules, and you need to know what are the energy consuming and the energy producing steps. We can go through it again individually in small groups, whatever it takes. This is highly, highly important that you learn it. I tried to go through and type out everything that I was saying so that you could understand it if you weren't getting what I was saying. Khan Academy has some really good videos on this process as well. There's a ton of good YouTube videos, I promise. You will get it. You're going to hate me for a while until you learn it, and then it'll make a whole bunch of sense. Okay? Something that I do want to point out is that if you start the process with glycogen, which we'll go over, it's another form of carbohydrate, if you will. It's another form of glucose. It's just a chain. It's the storage form of glucose. You don't need the initial energy input. So that first ATP input right here doesn't have to happen, because essentially instead of saying glucose, this would say glycogen. And you can actually go straight to glucose 6-phosphate from glycogen without the original energy investment step. So that means at the end, you would have 4 ATPs produced minus 1, because you have an energy input of 1 rather than of 2. So your total output, your net output of ATP from glycolysis, if you start with glycogen, is actually going to be 3. Weird caveat. Right now you're like, why is he telling me this? I promise you it's important. But you'll learn it. I have faith in you. So, aerobic energy metabolism. Why is it called aerobic energy metabolism? We're going to go over it. And we're going to go over the 2 processes that are actually involved in aerobic energy metabolism. One actually really doesn't require oxygen, and one doesn't use oxygen until the very last step. So it's somewhat annoying that they call it aerobic or oxygen dependent or oxidative phosphorylation. So where glycolysis ends with pyruvate or with lactate, but with pyruvate is where the Krebs cycle starts. So remember, glycolysis goes from a 6-carbon molecule of glucose down to 2 3-carbon molecules. If you look at the top, you can see that we have a 3-carbon molecule again. That's that same pyruvate. So really the primary goal of the first part of aerobic metabolism, or the Krebs cycle, or the citric acid cycle, or whatever you want to call it, is that we want to actually pull some electrons off and pull some hydrogens off and create NADH and FADH, because while ATP is great to produce in small amounts like you can with phosphocreatine or glycolysis or really with Krebs cycle, it's a lot more important to be able to produce high volume amounts of ATP. Excuse me. And that's exactly what NADH and FADH do. So these high energy NADHs and FADH are being created just to be exported and to be sent to a different spot to be used. And that's essentially, oxygen plays no role inside this process right here. It only plays a role in the final step of the electron transport chain, which we'll talk about. So here is the citric acid cycle. You start off with this pyruvate. Pyruvate gets transformed or converted into acetyl-CoA. That acetyl-CoA is what's going to be considered your hand stamp to get into Club Krebs. It sounds stupid to even say, but it's really what it is. It's your ticket through the door. It's your gateway. It's the most important conversion here in order for this cycle to run. Acetyl-CoA is a 2-carbon molecule. So what's going on when you go from pyruvate to acetyl-CoA? You're dropping off a carbon. That carbon's going somewhere else. Or really, any carbon-containing molecule can be turned into acetyl-CoA when we create this 2-carbon molecule. So we'll get into how you can burn fat through this process as well. What's kind of neat about this diagram is you can trace the carbons through. So you can see what carbons are original versus what carbons are being converted to CO2, and you can kind of see how they're shifting around inside of here. So the first real step of Krebs cycle is going to be turning acetyl-CoA into citrate, and then we're going to basically click around and around and around this process. And that's exactly, like, these are the exact same figures. One's just showing structure a little bit different, and one adds in the enzymes. So this one's actually adding in the enzymes for you to learn. So you've got acetyl-CoA right here. You're going to have citrate synthase catalyze the reaction from acetyl-CoA to citrate. So you're going from a 2-carbon molecule back to a 3-carbon molecule, right? Citrate is going to be transformed or converted into isocitrate via a conitase. Isocitrate is going to generate isocitrate to alpha-ketoglutarate via isocitrate dehydrogenase is really going to be the first kind of super important step here. This isocitrate dehydrogenase can be abbreviated IDH, and it's going to generate the first NADH out of Krebs cycle. After you get alpha-ketoglutarate and an NADH, you're going to use alpha-ketoglutarate dehydrogenase to convert alpha-ketoglutarate into succinyl-CoA. From succinyl-CoA, we're going to be converted into succinate via succinyl-CoA synthetase. And during this step, you're going to produce a GTP. GTP is eventually converted into ATP and thus is counted as ATP. From succinate, we're going to use succinate dehydrogenase to create fumarate. And fumarate is going to, this step from succinate to fumarate is going to generate our first FADH2, or FADH for short. This is another one of those high energy molecules that is going to be transporting electrons and hydrogen ions. Remember, the two major players in electron transport are NADH and FADH. So from fumarate, you're going to use the enzyme fumarase to get malate. And then from malate, you're going to generate oxaloacetate from the enzyme, or via the enzyme malate dehydrogenase. And again, here you get the generation of another NADH, so another electron hydrogen ion carrier. Yes, unfortunately, you need to learn the steps in the order, just because it will help you kind of track where these steps are coming in, and because in nutrition we will talk about each one of these enzymes and these intermediates, because different, like, things like protein come into play in the generation of some of these intermediates. So while we're not actually burning protein generally in exercise, we're using some proteins to generate some of these intermediates. So it sounds annoying, and truthfully, it kind of is. It kind of does suck that you have to memorize this stuff now, but it's super important to just memorize what the kind of reactants are, what the enzymes are, and what the products are. You also really need to learn what the steps are that are generating your NADHs, your FADHs, and your ATPs. If we're tracking through here, remember that out of glycolysis, you get 2 pyruvates. So essentially for every pyruvate, you get 1 click. So for each glucose molecule that gets broken down via glycolysis, you get 2 clicks of Krebs cycle. So that means that you get double everything that is produced here. So if you get 3 NADH and 1 FADH and 1 GTP or ATP per click or per run through Krebs cycle, you're actually per glucose molecule getting 6 NADH, 2 FADH, and 2 ATP because you're running it through twice. That's why it's so important to know that step where glycolysis splits into the glyceraldehyde 3-phosphate. Here, we're actually counting the ATPs that are going to be generated. It'll make more sense after you make it through this next section, but essentially for each one of these NADHs or FADHs, we're actually generating either 2.5 ATP or 1.5 ATP. And that's why they're so important, right? If one NADH can create almost 3 ATP, one NADH is making 2.5 ATP, and like one FADH is making 1.5 ATP. You, with one single NADH, you just made more ATP than this thing running through this entire Krebs cycle running through 2 times, right? Like it's so much higher potential energy, and it makes so much more ATP, and that's why it's important. Is that while it's taking a little bit more time, it's essentially like a 401k. It takes longer to accrue the money. But once you go to finally tap that nest egg, you're going to have a ton of money there. You know, you're kind of like sitting on it dormant and waiting and waiting and waiting. And as soon as it clicks over, you've got a ton of energy. And so here, what we're doing is we're counting the number of ATP. We're kind of tallying the number of ATP that are coming out of Krebs cycle. And you can see that you're adding up. So you're going, in glycolysis, you get 2 ATP net that are produced. You produce 4, you have 2 energy input, so you're getting 2 out if it's anaerobic. If you stop at glycolysis, if you have so essentially what I'm trying to say here is that for glycolysis, your net output is 2 ATP. 2 ATP is a subtotal. If you stop at glycolysis, hard stop, you're not moving forward, then you're only going to produce 2 ATP in general. However, if you're going to take and continue down the aerobic pathway and aerobic generation of ATP, you're going to create 5 ATP off of the 2 NADH that you produce, right? Because you need oxygen, you need the aerobic metabolism to be running in order to get the ATP off of the NADH. And so that's why I kind of drew these arrows here, is because it's super stupid the way that this textbook adds up stuff. But essentially you're going from right to left. 2 plus 5 is equal to 7. 7 ATP plus 5 ATP from NADH is going to create 12. 7 plus 5 is 12. And then 12 ATP plus the 2 GTP that you're going to make ends up with 14. 14 plus 15, 29. 29 plus 3 equals 32, right? It'll make more sense when we go through the next section, I promise. So how are we actually getting those ATP from NADH and FADH? Dr. Hyde, you keep saying this, but I don't know where it's coming from. Well, it's coming from the electron transport chain. Essentially what the electron transport chain is doing is it is serving as the little nest egg. It's serving as the 401k. It's serving as a massive amount of potential energy. And really it is NADH and FADH serving as hydrogen and electron waiters, if you will. Like they're working at a restaurant and all they're doing is they're serving food. They're servers. They're dropping off food. They're kind of taking the order in the first in glycolysis and in Krebs cycle, and then they're running that food back out, and people are getting to eat. They're literally dropping off hydrogens and dropping off electrons at the electron transport chain. That's what they're there for. As the electrons move down the electron transport chain, which I promise it makes more sense when you see it. As they move down the electron transport chain, their energy is going to be used to pump hydrogen ions up and out of the membrane. So they're actually going to be pumping hydrogens out of one membrane of the mitochondria and into a different space inside the mitochondria. So it's actually going to be like bottle rocketing these hydrogen ions away from the electrons and out. And then what you're going to end up getting is hydrogen ions coming careening down through a specific transporter inside of the membrane, clicking that over and creating ATP. Sounds super complicated. But when you have wonderful 8-year-old drawings, 8-year-old human beings, that is, drawing things, it looks a lot more simple. So what I want you to remember, or I want you to think about, is here's your phospholipid bilayer, right? You have these hydrophobic tails, and you have these hydrophilic heads. So you're actually inside the mitochondria, you have kind of like a cell inside of a cell. You actually have a very outermost membrane, and then you have a second innermost membrane. What you're doing with the electron transport chain is you're using an NADH as a waiter to come over and drop off an electron and a hydrogen ion. And there's actually 3 primary transporters or proton pumps, if you will, that are kind of jettisoning these hydrogens out. And they're going against their concentration gradient. So they're going, you're forcing more and more hydrogens into this area. And essentially, that's an area they don't want to go to. So you're using the energy from electrons to shoot them up and out and into this area they don't want to go to. So NADH is dropping off electrons and hydrogens at the first transporter. Those electrons travel down and travel down and travel down and continue jettisoning hydrogens up and out and up and out and up and out and up and out and up and out, right? FADH is going to be dropping off its hydrogen and its electrons at the second transporter, and that's going to be jettisoning out hydrogens as well. Whenever these hydrogens kind of continue around this intermembrane space, they'll reach a transporter called ATP synthase. And what ATP synthase does is it essentially controls the production of ATP. So because we have all these hydrogens outside in this intermembrane space in an area that they don't want to be in, they actually come careening down through ATP synthase and are essentially clicking over a turnstile like what you would see at a fair. And after every few clicks, it actually creates an ATP. So it's, as every hydrogen comes through, it's click, 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 ATP, click, 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 ATP, click, click, right? So every time it clicks, there's a hydrogen coming through, there's a hydrogen coming through, there's a hydrogen coming through. And then you're generating ATP. So where do we get those 2.5 and 1.5 numbers? So essentially what you're getting is for every Okay, so how do we get those numbers? Essentially, each one of these transporters, each one of these proton pumps takes some of the energy away from the electrons. So as the electrons move down, they become less powerful from here to here, less powerful from here to here, less powerful from here to here. So as they kind of continue on the electron transport chain, they're shooting out less protons, less hydrogen ions, less hydrogen ions, less hydrogen ions, less hydrogen ions. Because NADH is dropping off earlier, it's actually creating more ATP per unit of NADH. Because FADH is dropping off later, it's creating slightly less ATP per unit of FADH because NADH has one whole extra pump that it's able to get electrons and hydrogens pumped through. Does that make sense? So why is this considered oxidative? Why does it require oxygen? So if we did not have oxygen, then this pump right here would just continually collect electrons and continually collect electrons, because as they're moving down, this is where they're ending. This is where they're stopping. If we just forced this protein to become super negative, then it would no longer function because it would repel the other electrons. Electrons wouldn't be able to make it here. So what oxygen does is it's considered the final electron acceptor. Oxygen essentially snags away all of the used electrons from this final pump and combines with the hydrogens that are coming through ATP synthase to create H2O or water. So for every 2 hydrogens that come through, we get a water molecule that's being produced. Does that make sense? So it's super efficient. Just to give you an idea of where these hydrogens are being pumped, you see there's an outer membrane and there's an inner membrane. Those hydrogens are being pumped into this kind of darker yellow spot. So it's not like they're going outside the mitochondria. They're still technically within the mitochondria, but this on this border is where all of those pumps are and that ATP synthase is and all of that. Here's a little bit more explanation on how we get the numbers for those of you that are text readers and would prefer to read text instead of listen. Here's the pumps and the numbers that are associated with the text here. And this is essentially why hydrogen or why oxygen is considered the final electron acceptor. And it's this concept of pulling away the electrons like I just talked about. So all we've talked about is carbohydrate. All we've talked about is you put sugar in, you put glucose in, glucose eventually creates pyruvate, and pyruvate goes into Krebs cycle, and then you continue on to electron transport chain, right? So essentially, fats can also play into the Krebs cycle. Fats can play into this concept of aerobic metabolism or aerobic energy production, because those fats are just strings of carbon. And all you have to do is liberate those strings of carbon, and you have essentially an infinite number of acetyl-CoA that you can create. Because remember, acetyl-CoA is 2 carbon segments. So if a fat chain is a very, very, very long chain of carbons, every 2 carbons you cleave off, and it's 2 carbons, and it goes in and you can have it become acetyl-CoA and get fed into Krebs. And 2 carbons, acetyl-CoA, feed into Krebs. 2 carbons, acetyl-CoA, and feed into Krebs. For every 1 glucose molecule, you only get 2 pyruvate and 2 acetyl-CoA. Right? So it's very finite per glucose molecule. Per fat molecule, it's virtually limitless. I mean, as long as that chain is, you can create acetyl-CoA. It's a much more robust way of creating energy. It takes a little bit longer, but you can generate significantly more ATP from fat than you can from carbohydrate ever. Sorry. And then protein. Protein can actually contribute to become acetyl-CoA. It's very, very little amounts. Protein generally doesn't directly contribute to energy metabolism. It's not really producing energy. Instead, that protein is being converted into intermediates inside of Krebs cycle. So we can actually use some of the amino acids to create different intermediates inside of Krebs cycle. And those intermediates, just to remind you, are these. So like you can use protein to create some of these intermediates inside of Krebs cycle. So if you start running Krebs cycle super fast or very often, you're going to be limited by how many of these intermediates occur. So if you run out of any one of these intermediates, you're not going to be able to continue to click over Krebs cycle. It's going to get ended. So protein can actually come in and fill in some of the gaps, if you will. So realistically, how is all of this controlled? You've been talking a lot, Dr. Ide. You haven't really shared how we're controlling this. Some of it is really, to be honest, it's kind of using a little bit of logic. So what this table here is doing is it's showing you what are some of the stimulators and some of the inhibitors of what's called a rate-limiting enzyme. Rate-limiting enzymes, I didn't even cover it on accident, but essentially rate-limiting enzymes are the enzymes within a pathway that control really is the pathway going to continue on or not. They control the rate at which the pathway continues on. So in the phosphagen system, the rate-limiting enzyme is creatine kinase. If there's not enough creatine kinase, that reaction is not occurring, right? We're not going to create ATP via creatine kinase. For glycolysis, the rate-limiting enzyme is phosphofructokinase, or PFK. You need to know that. You need to memorize where that is in the chain of glycolysis, and you need to know that phosphofructokinase is the rate-limiting enzyme. For Krebs cycle, it's isocitrate dehydrogenase, and for the electron transport chain, it's cytochrome oxidase. It's one of those pumps, basically. The stimulators of those rate-limiting enzymes, so things that make them more active, things that make them want to create more energy, things that make them want to go at a faster speed, are called stimulators. And really what those things that stimulate the rate-limiting enzymes are low energy sensors. So ATP is a form of high energy, right? ADP means that ATP has been used and has created ADP. Kind of circular, but essentially ADP is the breakdown product of ATP. If we've broken down ATP, that means we've used energy, and that means we need to create more energy. AMP is adenosine monophosphate, and that's actually the breakdown product of ADP. So if we use ATP and then we need energy, so we use ADP, we create AMP, right? So AMP is like the you officially hit the low fuel light, right? Like you're now playing games with whether or not you're going to be pushing your truck or not. AMP will stimulate these processes to speed up as well. On the other side, what is inhibiting these pathways? What's slowing them down? What's putting the brakes on them? And that's going to be markers of high energy availability. So that means if ATP is present in high amounts, why do we care? We don't need to create more ATP. We're good. We're good. We've got ATP. We've got energy. We don't need any more, right? ATP, high energy, will literally symbolize and signal the stop of some of these programs, the stop of some of these pathways. And that's the same thing with NADH. NADH will actually stop some of these pathways from continuing on because it'll say, hey, listen, we've got all the potential energy that we need. We don't need any more. There's no need to waste time with this. We're good, man. Okay. I hope that makes sense. If it doesn't, please reach out to me. I'm more than happy to sit down in small groups. I'm more than happy to sit down one-on-one, go through Zoom calls, whatever it takes. This stuff is super important, and really, you're kind of at a slight disadvantage because we can't sit down in class and draw this out and ask questions. So please use me. Please. No.